Right. Well, thank you. Thank you for being here. I hope I can keep up with pizza as a target of interest during lunchtime. So yes, I am coming to you from the School of Arts, Humanities, and Social Sciences. The program in Science, Technology, and Society is an interdisciplinary program that combines aspects of history, anthropology, sociology, dash of economics from time to time. And it's one of the reasons that I love working there is you get to take big issues, big ideas in science and technology and think about them from a variety of perspectives. And I say that by way of prefacing the talk I'm going to give you, which is going to use history in principle to explore what are much more contemporary questions that you might find interesting about biomanufacturing, which of course is closely associated with the bioeconomy. One of the reasons you might want to think historically about this topic is that there's really not much discussion of biomanufacturing per se, as imperfect as Google Ngrams may be for tracing word use over time, until roughly 1979. I promise you there's a little blip right before 1980. So biomanufacturing is an activity in history in search of a name for most of its duration. So we can't actually find examples of biomanufacturing as such if we go back into the early 20th century or before. And if you're trying to think about the nature of what makes an industrial revolution, what makes a manufacturing revolution, of course, you may want to be able to identify instances of it. This is a classic issue that historians face dealing with concepts before the term. Incidentally, of course, the only reason we have biomanufacturing as a term is largely thanks to NASA and Skylab in the late 70s. That's the blip that I mentioned earlier. NASA is very interested in biomanufacturing, not as a substitute for the conventional industrial petro-fueled economy at the time, but as a matter of how can you produce things in space under very different resource constraints. And so biomanufacturing starts there, and then as I've been tracing the term, propagates out. That's a conversation for another time. Coming back more practically, this is a definition from the Biden administration's bioeconomy program. You see a stress on biomanufacturing as the use of biological systems, including plants and microbes such as bacteria, yeast, and algae, or their components to produce products at relevant commercial scale. And then they say, for example, since the 1980s. So my sort of complaint and my observation here is that the history of biomanufacturing as a term and how it's showing up in current policy discussions is fairly truncated. 1980 only gets you back about nearly 45 years. It's a lot of times that we've been doing what sounds like this activity, using biological systems, including plants and microbes, to produce things that are useful to us at a commercial scale. You can think about the production of alcohol, the production of fibers through both plant and animal husbandry, the production of food. All of these things might seem to fit within a more capacious understanding of biomanufacturing, but typically when you see timelines associated with the biomanufacturing revolution or other sort of appellations, I don't mean to pick on this particular author, I just sort of grabbed it because it's the most convenient image you see. Yep, it's intentionally truncated. Oh. Yeah, don't worry, I didn't want to, yeah. You can see, oh yeah, sure. Oh. Okay. This is the full screen, as we can manage. It's working within my browser. Sorry about that. This is doing Google Slides inside of a screen share. So what I want to emphasize here is BM, which is their acronym for biological manufacturing. It's usually, or biomanufacturing is usually one word, so it could just be B, I guess. But it has, a version in the 1910s, a version in the 1940s, a version in the 1980s, and the 2000s. We'll talk about all of these in time. But this type of diagram shows them as being very discrete events. You have one and then you have the other. And one of the insights from the history of technology is that things don't happen and then stop. They keep going. So for example, during the Industrial Revolution, which is famous for the use of steam and later petrochemical power, the highest use of horsepower actually occurs in the 1950s. So up until then, you have more and more animal power being used every year. I mean, famously, of course, the armies of the First and Second World Wars were largely run on horsepower as opposed to mechanization. So that's, if you're trying to understand, say, how manufacturing done and how the social activities are being conducted, it doesn't really help you to say there's this new thing and this new thing alone. This is always going to coexist with other forms of what historians of technology call reserve technology. There's always an alternative out there. The thing you were doing before is still there as a possibility. And so if you're thinking about sort of this idea of a revolution, it doesn't quite capture the historical process to which these new ideas come in, because it's never only asking, is it possible, is it desirable, is it possible or desirable in relationship to what? In relationship to what set of social needs, in relationship to what set of social structures have been responsible for production. And it's with that insight that I'm now going to sort of reach farther back in time and take you into sort of my most polemical part of the talk, which is going to be sort of saying that there's biomanufacturing as such could be thought of as being something that we can think of in the history of agriculture. And indeed, the history of agriculture gives us a number of important cues for how we might think about the social implications and operations of biomanufacturing today. And then through the lens of the development of biotechnology, I'll sort of take you on a tour of sort of these sort of first three moments, at least, in contemporary biomanufacturing, explain how they fit into that schema. So with that, why am I showing you a picture of this cow? This may not look like it, but this is a picture of advanced biotechnology in the early 19th century. And I'll give you a moment to pause to think of as to why. Are there any reasons why you might think of this cow as being especially advanced? It's a very large cow, right? So depending on what you want to do with your animals, if you want to use them for motive power or meat, you will design different animals. And this is an example of being able to build a very large animal, which will be useful towards that front. And this is an example of there's this huge discussion, which Darwin, for example, draws very heavily upon in the 19th century before, around raising and breeding animals to meet certain economic needs. And what I want to emphasize here is that often if you think about the idea of controlling life, you immediately, of course, we're at MIT, you think of various forms of engineering. You probably think about genetics, the ability to see and manipulate things at the molecular level as a precursor to sort of dramatically transforming life. But actually, human societies have been transforming life in very dramatic ways to suit their own ends, independent of a knowledge of what's going on at that basic level. And agriculture provides numerous examples. I'm just going to dwell on a few of them. And in particular, I'm going to dwell on sheep in Europe and England in the sort of late medieval to early industrial revolution. Sheep were, of course, you can always eat a sheep, but the original sheep that Europeans raised were often raised through transhumanance, which is being kept up in the mountains for part of the year, brought down to the coastal plain during the winter, and then back up. They do a lot of walking. It's a pastoral process of grazing, very muscly, very stringy. Mutton is not something you especially want to eat if you're looking for a meat to eat, but they make this wonderful wool. And sheep are bred to produce that fiber, which is incredibly valuable. It's one of the most valuable exports that England has during the late medieval era. They're shipping that off to places like Flanders, where it's being woven into wonderful tapestries. And so England has a huge sheep population that looks a lot like this kind of sheep, in the sense that it's very bushy. It's got a lot of wool because that's the part of the sheep that's important, and that's the part that's maxed out. However, with time, England begins to change. Markets for wool begin to change. By the late 18th century, you have rapid industrialization. You have many more people no longer living close to food on farms. They're living in cities. Those people need to feed themselves. They're very hungry. There's suddenly this huge domestic market for forms of meat. And that's something that a British agriculturalist who has a wonderful term of grazier in the 18th century named Robert Bakewell discovers. And I'm going to dwell on Bakewell because he is the most famous and notorious animal breeder in the Anglophone world in the 18th century. In part, it's for his methods. He ends up using quite a bit of what's considered incest to breed his sheep, because he called breeding in within. But he is very interested in this project. How do you take the practice of raising sheep, which typically involves them just wandering on pastures and growing wool, and how do you make that process more efficient? And part of that, of course, is how he runs the land that he owns and he rents. He sets up these very precise paddocks that have equal amounts of sort of forage and water. He keeps very careful records of the lineages of his sheep, and as they're bred through successive generations, to sort of track the properties that he associates with given lines. So this is all, and he makes sure that he irrigates his land. So these are all things that he does that we would sort of recognize as intensive agriculture, but they're also forms, as he understands it, controlling the form that his sheep will take. And he is remarkably successful in the course of, a few generations worth of sheep. He begins to produce what becomes known as the dishy sheep. which is a very different kind of sheep than existed in England a few generations before. I'm not sure if, once again, keep in mind if this is the sheep that you would want for meat, what are some of the notable features that have like maximized your profit as a grazier? Yeah, there's very skinny legs, no horns, this is a ram, so it should, when am I expected to have horns? And a very short coat. You basically, through breeding, he sort of removed as many parts of the sheep that were extraneous to the production of muscle as he reasonably could, and more or less create a meat cube on legs. And he does this very rapidly within the scale of an animal breed. His findings are widely discussed by the Royal Society. For those of you, I assume are many in here who are familiar with Charles Darwin. Charles Darwin's mechanisms for natural selection are heavily conditioned by his observation of English breeders and the tremendous plasticity of life under their control. So this is, once again, if you're thinking about biomanufacturing as manipulating living systems to produce desired products at scale, this type of animal husbandry has a reasonable claim to be included in that history. Although, and once again, you see many antecedents here of sort of factory farming and industrialized agriculture, in the sense he's trying to take a process, and agriculture, of course, is a famously capricious and unpredictable process, and turn it into something that's rational and profitable, which sounds, once again, quite a bit like the promise of many forms of biomanufacturing today. And it pops up in the frameworks of more famous contemporary innovators well before he was in charge of a new branch of the federal government. Elon Musk was famous for promoting one of the many sheep breeds that the highlighted at the Museum of English Rural Life. So, and once again, this is a sheep that's not for meat, but another type of once it goes to how quickly you can change these breeds. And this, of course, is a venerated, famous, and honored pastime in agricultural husbandry. There are, however, forms of agricultural innovation which are less fuzzy, if you will, and more hard. And I'm talking, of course, about the development of hard wheat and its spread. So this is from a group of economic geographers, and it shows two maps. The first is where wheat production is in the 1830s in the United States, and the second one beneath is wheat production in sort of about 50 years later. And it might just be the story here that there's more of the United States that's been incorporated and settled, but in this interval, the geographic center of where wheat is being cultivated in the United States moves about 1,500 miles. And it changes climatic zones dramatically. There's a big difference when the center of your cultivation is, say, the Ohio River Valley, which is very moist and generally wet and warm, to where wheat becomes very well cultivated in the United States, which is on the plains of the Dakotas, where temperatures are very extreme. It can be much drier. So here, the real is sort of a type of Red Queen situation, where the real triumph here is moving is standing still, although you're actually moving. And this is the product of a concerted effort by the United States Department of Agriculture to collect wheat germplasm from around the world and identify the strains that are best suited to the climate where it's happening. So once again, the type of wheat which is being developed is with the aim of enabling agricultural expansion. Once again, it shows how quickly you can begin to sort of change the nature of a plant to enable your overall progress. So sometimes these types of innovation, this is sort of the overall play, sometimes they're very obvious, and sometimes they can be very subtle in terms of things that simply continue to happen, even as everything around them changes. There's once again sort of a way of calibrating our sensibilities of where to look for meaningful events. And in general, this is true of plant breeding more generally. The late 19th century, the idea of changing the nature of fruits and flowers is something that's seen as terribly innovative and technological. This is once again in a pre-Mendelian genetics era, but nonetheless, the triumphs and abilities of various breeders, such as Burbank, are widely discussed and lauded. Indeed, Burbank is profiled as being alongside Thomas Edison and Henry Ford. So once again, these two titans of mass production and electrical engineering being paired with somebody whose main accomplishment is making sort of new forms of apples and fruits and berries gives you a sense of the esteem in which this type of work was being done, once again, prior to genetics as we formally understand it. So you don't need the science necessarily to have this sort of reputation for being able to make progress and having innovations. So it suggests that maybe some of the technological innovations that are presented as a reason for novelty can be, read against this broader set of thoughts. And that's especially true when you think about other famous agricultural innovations in the early 20th century, such as hybrid corn. Is that it's one of the foundations of the Green Revolution. And this is, of course, is a story where this is not about, it's about what we value, but often what we're not told is an actor in the story about what makes for a successful new technology. Hybrid corn, of course, comes from the theory of hybrid vigor that's been observed by breeders for quite some time, which is that if you you cross things that are slightly different, you often get progeny that are more exceptional in some traits. And people had spent quite a bit of time studying corn in this manner, sort of year by year. And this breeding of corn is a very complicated, laborious process, especially if you want to do hybrid corn, because you have to detassel every single ear of corn in the field, and you have to worry about wind spread. So for a long time, it seemed as if hybrid corn was not going to be especially remunerative because of the labor that went into it. However, in the 1930s, seed corn for hybrid corn becomes very popular. And it becomes popular for two reasons. One reason, of course, is that you can actually sell it. You can't sell normal corn varieties because they continue to grow like plants do, whereas hybrid corn, if done properly, only has one generation, so you have to continually get more seeds. That's the reason as an agricultural seed seller, you might want to investigate this type of hybridization. The other big reason you're often told is that hybrid cornfields are more productive. You get more bushels of corn per hectare from hybrid corn than other corn. Who wouldn't want more corn? Who wouldn't want more food? The surprising thing, though, of course, is that this moment of hybrid corn adaptation occurs in the middle of in American farms, in the middle of the Great Depression, when commodity prices are at an all-time low. When, in fact, farmers are throwing away or burning or otherwise destroying crops because it's not worth the money to harvest them and bring them to market. So why in that moment is it worthwhile to have more of something that nobody wants to pay you money for? And the historian's answer, at least, is this is also the era of the New Deal, of the Agricultural Adjustment Administration, of the Farm Bureau, and the response that they had to the excess, having too much food, actually, or at least too much food for what the market considered worth paying for, was to try to restrict agricultural production. In essence, you would go to farmers. This is still a widespread practice. You go to farmers and say, "We're going to pay you money not to grow. We see you could grow this much on your field. We're going to pay you to not grow anything on that field. We're going to trade you the assured income of a government subsidy so that you're not flooding the market with all the stuff you feel obligated to try to grow and driving prices down for everybody else." This is a long-standing issue for people for organized agriculture. So in that context, the reason that hybrid seed corn is so popular in the New Deal is because farmers figure out relatively quickly that you can sort of have the best of all worlds. You can get paid money to take part of your land out of circulation, but you can still grow the same amount of corn that you were before in a smaller amount of land. So as farmers see it, that's a good deal because you're getting paid money for the land you aren't using, and this new hybrid strain allows you to produce what you were producing before. So the greater vicinity of hybrid corn is certainly a part of why it's successful, but it's not necessarily in the way that, say, addresses a problem such as a resource shortage or world hunger. And there are, of course, many points in the manufacturing of biological things and other things that are, if you poke into their social relations, are full of sort of these perverse or unexpected outcomes. And that's sort of the big lesson that I sort of want to give you coming out of this part on agriculture, because I doubt none of you are going to be detasseling corn, or I could be wrong. I'm not sure if you're going to be breeding sheep, although an MIT sheep would be kind of interesting. But that said, these basic ideas that there are changing social and market priorities that can affect how you're trying to reshape living organisms, and that people have been doing some version of this for quite a while, I think is an important lesson to take into how we think about the potential future social relations of biomanufacturing. Which brings me to some of the more familiar forms of biomanufacturing that you may have walked into this room today to think about. This, of course, is not contemporary biomanufacturing. This is taken in the Middle East. It represents some of the first wells used for fermentation about 13,000 years ago. So, of course, fermentation is a core biomanufacturing process in certain ways. You cultivate these microorganisms in your milk and your in your sugars, and you get something much more interesting out and much more useful. So this is a process that's been going on for some time, but in the late 19th century, it becomes sort of rationalized through science and sort of become the basis of the idea that you could use biological systems as a substitute for some of the things that we're seeing as only coming from what I'll broadly call the physical world, that ends up being very closely associated with the carbon economy. And we owe a lot of this to Louis Pasteur, who I'm sure is a familiar name to all of you here, for his contributions to microbiology, for pasteurization, for his work on vaccines. But Pasteur's interest in microbial life, you may not know, actually emerged out of an intense theological and political debate in 19th century France. His first experiments demonstrating that sort of life could only come from life, that you need microbes to get other microbes, emerge in response to a long-running debate over spontaneous generation. And the question of spontaneous generation for French debates in the 19th century has a very clear theological valence. Because if you are, say, a socialist and a scientist, you wanted to say that there's no special thing about matter. Right? Life is just matter in a different form, which means that spontaneous generation, the emergence of life from inanimate matter, should totally be possible. And in fact, the fact that it's possible also, as a theological bank shot, means you don't need a first mover. Even within the Christian theological tradition, you don't need the creation of life at a given time. So actually, spontaneous generation is one of those moments, much like debates over evolution somewhat later, where there's a suggestion you don't need a creator to sort of guide the evolution or operation or emergence of life. So this is a very heated question. And Louis Pasteur uses his knowledge of emerging knowledge of microbiology to actually say, take sides with sort of more conservative religious aspects of French society, because he says quite deliberately, no, you only get life from life. And the way he proves it is through developing and perfecting a number of the sterilization techniques and ideas premised on the ubiquity of microbiological life everywhere. And so this shows some of the goose and thin-necked flasks that he used to sort of for these experiments. He put like a kind of broth in these flasks. He would then sort of evacuate the air as best he could, and then sort of melt the top and then seal it. And he showed that if you actually were appropriate about sterilizing these, they just remained inert. And therefore, any other proofs of spontaneous generation were the result of microbial contamination. But all of this work on identifying what kinds of microbes are where, how to create sterile environments for other things to live, is, of course, very well adapted to making fermentation a more effective process. I mean, people have been making wine and beer and other for quite some time, but it's a hard process. You need to know you're feeding the right bacteria. You want to keep your systems clean. Things go wrong. You don't quite understand why. So this is a case where having new microbiological knowledge is very helpful for making fermentation a more effective system. Pasteur is very much a French nationalist. He wants to make beer better than the Germans. He wants to make French wine that much better. So his study of microbiology is actually very much entwined with his interest in sort of promoting France through assisting what we would recognize as its biomanufacturing efforts. And there's just taking the new ideas, like, can you observe the process of fermentation without opening the tank? And if you build windows, these are all things that are premised on the presence of microbes that you wouldn't necessarily have in fermentation technology were it not for that decision. And that's sort of the scale, which when I talk to historians, I have to convince them that this is literally a big process, but I probably don't have to do as much work for all of you. That brings us to a different moment, which is often associated, I guess nationalism, German-French rivalry is very much the point. This is a picture of a battlefield in the Western Front of the First World War, probably in northern France. And it has a few features which are striking, but the main one that I want to highlight is barbed wire, which is everywhere. In this picture, it was everywhere on the Western Front. It was a product which had been developed for controlling ranges in the American West by keeping cattle bound in. But you take barbed wire and you put it together with new inventions such as the machine gun, you create a lethal barrier to advancing infantry. This is, of course, characterized, I'm sure this is familiar to you from your thumbnail understanding of the First World War. The thing that you may not be aware of is that most of the artillery shells that were used by the Allied forces in the first part of the war could not cut barbed wire. The types of shrapnel that were produced and the angles at which they went out would rarely toss the barbed wire around. They would never shred it or destroy it. They could not really displace it. So you would have battles that were characterized by massive artillery bombardments. And then, of course, the soldiers would go forward and get caught in barbed wire and slaughtered by the German defenders because the barbed wire had not been shredded. There was a way to use explosives to destroy barbed wire using high explosives. The problem was that high explosives depended very heavily on a compound called acetone. And in 1914, 1915, the principal producer of acetone was Germany, using much of the German sort of pharmaceutical and chemical industry in the late 19th century. Like, what can we do with coal and its byproducts? It turns out, so basically producing acetone out of coal and sort of the compounds you get from working with coal, which of course the British and the French don't have. So they are really, really desperate to find a new source of acetone, which is where Chaim Weizmann comes in, who's someone who's been working in industrial fermentation. He is the person who's credited often with starting to become systematic. Like, we know that yeast is really helpful for making bread and booze, but like, what other types of microbes might produce useful substances? And by the 1916, 1917, he's figured out that there's a Clostridium axio, sorry, I apologize for my throat. Axiobacillium is actually very helpful, as its the second name suggests, in making acetone or its precursors. And so you can produce acetone via the fermentation of carbohydrates. And this, of course, is the type of diagram you may be familiar with from your early customs. As the acetone and butanol cycle is which he sells to the British the government, becomes the basis for vastly increasing explosive production, and of course, plays a part in helping the Allies breach German lines and secure victory. But for the purposes of this talk, the very important thing is he said, "Oh, actually, all these things that you thought of as being produced out of coal or petroleum products, maybe there's a biological pathway that could be carried out." And this is just an example of one of the fermentation rooms for the acetyl-butanol system in Canada, where there's a lot of carbohydrates. Because suddenly you go to places with a lot of agricultural products and you start topping them in with your bacteria. And that's so it becomes produced on an industrial scale. And then in sort of this early 20th century time, with the emergence of this sort of first round of sort of biomanufacturing beyond fermentation, you see a proliferation of terms, not of biomanufacturing, but of biotechnology. And I'm only going to mention them here briefly because often when you think about biomanufacturing, it's based on some biotechnological platform. And the question is, who's defining it, who's thinking about it? And you have folks like a German thinker named Gustav Tornier, who says, very much a definition that might resonate with biological engineering today in terms of you study how they're built up according to physiotechnical laws. So there's nothing special about life. You can sort of study it the same way you study matter. You then have biotechnology, which is a French term, which no one's ever heard of R.H. France anymore. But back in the 1920s, his books were widely read, and he sort of once again sort of popularized this idea of a biologically based technology for Francophone audience. But one of the most famous uses of biotechnology actually comes from a pig farmer in the former Austro-Hungarian Empire named Karl Erke, who coins this term. And it's based on his explanation of the special methods, once again, that he uses for cultivating pigs. So one of the reasons I wanted to start with agriculture is you do have people in the early 20th century trying to make sense of biotechnology through the agriculture and sort of the scientific by this point not only means sort of organization, but also means something like being able to take account of like nutritional inputs to how you contain and raise animals. In terms of this idea of revolutionary areas, you have Patrick Geddes, who's well known for his work on urban theory, but he says that potentially humankind is entering a biotechnic era, although once again, I have sort of the same question for him as I do for the Biden White House, which is what makes the activity you're describing new. And if you're only concerned about using biological organisms to benefit mankind, because that sounds like quite a bit of human history, at least as I understand it. And this provides the context for, there's a lot of work on fermentation as a production system for lots of different chemicals, but it gets an additional boost in the Second World War with the discovery of penicillin as the antimicrobial byproduct. And this, of course, is once again producing these large-scale fermentation efforts. If you are in the history of medicine class, they'll just tell you about the discovery of penicillin by Alexander Fleming. They don't usually dwell on the types of technological innovation it took to manufacture penicillin at scale quite rapidly during the war. But that's really the success story in many ways, right? It doesn't matter if you have a discovery; it needs to be scalable. And then there's sort of this hinge point. This is like why, as a historian, I sort of cast such a far net back. With the rise of conventional, molecular biology, you begin to see people within that community, within biochemistry and molecular biology, beginning to talk about engineering or designing life from a very different perspective, one that really highlights their own expertise in the science of life, as opposed to all these other ways that people had of manipulating life. So this is Edward Tatum, who's famous for the one gene, one enzyme hypothesis. He says in 1958, proposed will be able to figure out this code of life processes that will permit our improvement of all living organisms by processes which we might call biological engineering. So we can maybe talk a little bit about what biological engineering means to you, but this is where it's very much located at the molecular level. It suggests that there's sort of a small scale way of doing this that will have large scale effects, as opposed to the types of what I would say might pass for biological engineering through husbandry and agriculture, which are very much macro scale. This, of course, converges with a much longer ranging debate among engineers and scientists during the Vietnam War about what ends their research will be put towards. And there's a huge amount of interest in using new engineering and physics methods, starting with people recoiling from the atomic bomb project and then going into sort of concerns about their complicity with the US military policy to research life instead of researching death. And so you have a huge flood of interest in the possibility that researching life at the molecular level will produce all these beneficial advances to humankind. And that famously produces the discovery of recombinant DNA in the early 1970s, which of course, speaking of social relations, is also patented as a method of doing research. And it's initially seen as a method of doing research. You want to understand how a gene works in the early 1970s. You can't sequence it. You don't know how to sequence things. What you do is you try to slice it up with the limited number of enzymes you have, and you insert that chunk back into a bacteria like E. coli, and you see what it does. Incidentally, in my prior work on cancer viruses, I found out this was very controversial because one of the first systems they did this with were tumor viruses, which happened to have well-defined small genomes, and people got nervous about swapping tumor virus DNA into bacteria. That's my second book. So for this, though, it might have started as a way of studying life by isolating genes, but this also sounds like a really good way of producing very complicated molecules. You can find the gene responsible for, say, producing insulin, and you don't have to worry about getting a pig to produce it for you in their pancreas and then killing the pig and extracting the insulin, which is one of the ways that insulin was being produced for people who needed to treat diabetes and other conditions. You can just swap that gene as a plasmid into E. coli, and E. coli will quite happily grow for much less effort than it takes to get a pig to grow, and it will produce this as a byproduct. Before I go on to that next slide, you can see somewhat of the concerns, like the technical name for these new E. coli are chimeras, which of course is a Greek term for sort of a mythical, many-headed, many-parted beast. So there's that idea is still drifts around beneath the rhetoric of this. So this is where sort of the current interest in biomanufacturing might be rooted. This is once again 1979, that magic year in which a venture capital firm named E.F. Hutton holds a seminar called DNA, the Genetic Revolution, in which they sort of lay forth this idea that I've said as a business plan, which is you could find these genes and use them to produce things like insulin, possibly growth hormones, other things that are proteins of therapeutic value, much more cheaply than you'd ever be able to do them through these other methods. Once again, there are other methods. It is possible to get insulin by going to a slaughterhouse and purifying the extracts of what you find there. But this is going to be much cheaper, much more lucrative, much more valuable because it's tied into that market for medical care. And indeed, as the founder of one of the founders of Genentech and promoters said, we should all be lucky to have bacteria, workers who work for us as hard as bacteria do. They will work 24 hours a day, 7 days a week making your product. And moreover, they'll die to do it for you. And so that's sort of the pitch, and that's the feeling of the excitement over this new form of biotechnological manufacturing of pharmaceuticals in the early 1980s. And you see Genentech is sort of the first and most famous exemplar of this, although South San Francisco makes any number of claims I would take issue with about the origins of biotechnology and its self-promotion. And this is one of the fermenters that Genentech used to do it. They're now preserved at the National Museum of American History down in Washington, DC, but it's still pretty small. Like, you don't have a sense of scale from this diagram, but it's a little bit bigger than a benchtop unit that you might use. So it's bigger than what you might produce in the lab, but it's still not this sort of vast fermentation vats that we saw for the acetone butanol process or for brewing beer. But these are the types of devices, and that's the critical link. It's not just enough to have genetic code, you also need to have a means of manufacturing through these older processes. So that the merger of genetics and this older technology of fermentation is what makes the biotechnological revolution that's promised possible. So it's not just a new thing displacing the old, it's reusing it. Much in the same way, I guess you can say the slaughterhouses are a creative reappropriation of the process of husbandry to make useful compounds. It also plays out closer to home. People are familiar with this building. Does anyone know what this building is? You go up towards Harvard on Memorial Drive. It used to be Genzyme, yeah. And not only is it Genzyme is there, it was the manufacturing facility they built in the early 90s to produce the recombinant version of their most famous and first drug, cedarase, which treats a metabolic condition known as Gaucher's disease. This is, of course, a very familiar early biotech story, which is that you can isolate the enzyme that people aren't producing when they have the condition, and you give it to them as a supplement to reverse the effects of the condition. But the only source commercially available of the enzyme in the late 1970s and early 1980s was the placentas of recently delivered human beings. And so Genzyme's first effort, which was successful, was to contract with a hospital in France that would collect untold thousands of these placentas and purify them to the state where they could be flown to one of Genzyme's manufacturing facilities and further purified and handed out. This is what made cedarase, when it was produced, one of the most expensive drugs in the world, but also was life-changing for people who had Gaucher's disease. But Genzyme didn't feel it could purify placentas forever, so they were trying to develop a recombinant main, which is finding the gene responsible for the enzyme and then producing it through fermentation. And that's what this red brick plant was intended to do. That's what it did do with the product cedarase. But it's also the site of Genzyme's downfall, because several years later, somebody doesn't quite do their job cleaning out a fermentation tank, and the process of production is contaminated. And because it's both a pharmaceutical and it's a delicate process, the financial hit they take from it in the biotech process, regardless of being the reason that they had to eventually merge with Sanofi and sell out. And so these, once again, these very, they're focused on molecular biology and genes, but like these older questions of microbial motility and contamination will sort of pop up always when you're thinking about, so they still have that level of their activity. And that's really, I guess, where I'll leave us for now. I mean, you can see many more promises around Boston, places like Ginkgo Bioworks, et cetera, making the organism that's right for your job. This idea, once again, that life could be dramatically reshaped to serve whatever needs you need, to sort of in a manufacturing sense, which of course is very exciting. But I don't think I can sort of say that it is novel. And of course, as a historian, that's what I want to leave you with. Like, potentially, as you're thinking about this, or who knows what technologies you're going to come out, but in your time at MIT and beyond, that there are going to be some points of interesting continuity that we're thinking about, even as you're witnessing immense technical change. So I'm looking forward to having a few questions in the remainder of the hour. Thank you so much.
We have a few minutes for everyone. So maybe we'll pass the mic. I think I can yell. Yeah, I don't want to. That was an excellent talk. Very and it's like you emphasized a couple of times definitions and nomenclature. And I think by really good historians, but I know there's like a lot of different economy. Could you talk a little bit about why it's important to have agreement about what we call these things that we do?
Yeah, this is a historian of science. Science is one of the things we enjoy most. We enjoy sort of talking about how things are talked about. But it does matter. And to give a semi-biographical example, I started becoming interested in the bioeconomy, maybe 8 years ago when I was working on this sort of biopharmaceutical project. And it took sort of running into Mark being this year for me to realize that the bioeconomy is actually much more than just being sort of like the impact of like pharmaceutical manufacturing. And it actually has these other branches, and it has these that's like partially how you name it involves where you go looking for it. And certainly at the level of policy, that's why these definitions are so important, because depending on how you define it, that will shape what types of projects get funding, what types of impacts and stakeholders sort of become interested in promoting it. And then I think I can say this. So I'm hopefully not going to be on a panel at the National Academy of Sciences for talking about engineering biology. And like the first question they asked me when I was interviewing is like, what's the bioeconomy? Because they're trying to figure it out. And they're figuring it out too with an eye on politics. And they wanted to know, actually, ironically, how much I could talk about the rural impact of the bioeconomy. Because they're sort of thinking, all these things map onto those concerns that people bring, but at the same time, they feed forward then into the types of research that will get funded or not funded under that idea, and how people think that their own work is relating to it. So that would be my brief answer. But those are both the conceptual and practical reasons to think about definitions in the present day as well.
You mentioned the pursuits that these sort of plant between people and animals and people doing. I feel like we kind of see that today as well with a lot of academic scientists focusing on strain engineering. But the cultivation technologies, like things like bioreactors and the technologies that go into like conventional agriculture, also arguably make a really big impact in the productivity. Can you speak to like how the prestige in conventional agriculture sort of compared between the people producing strains versus the technologies that are harvesting?
Yeah, that's so I mean, I will say that I was listening to Professor Laufenberger talk yesterday at the Heels launch, and he had this point that like there's all this technology relevant to biology that, and I'm speaking as a historian of molecular biology initially, I'm completely guilty of that initially, and I'm trying to think my way through it. So I would say that if you actually, there's a former emeritus professor from my program, Deborah Fitzgerald, who wrote a book called Every Farm a Factory, where she talks very deeply about the types of combines and harvesters and sort of the mechanization as an important side. That was actually what was being exported to, say, the Soviet Union when they were trying to modernize. They were deeply interested in sort of those types of technologies. So in that sense, you see quite a bit of discussion. If you think about if you go to Chicago, you sort of look at McCormick Place, sort of the McCormick Reaper, like there's a lot of that is sort of is a lot of, I don't quite, you don't have and actually McCormick might have been a candidate for like one of those photos of like great innovators in the early 20th century, although he wasn't there. So I think that would be one of the places I would look in. Today, I think that's very much the sort of the scene that, one of the points of contact with biological engineering and other forms of the biological sciences, which is that how do you take something that's interesting and make it practical, and how to do it. And that's a process that people often skip over. So if you go looking for biomanufacturing, you'll find something periodized only by scientific ideas, which doesn't really, in my mind, give you a decent grasp of what's actually going on both in the moment or over the course of its development. Mm-hmm. Yeah, so I'll only say the reason I started there is because I had 45 minutes and I didn't have an hour and a half. There's a lot of really interesting stuff that people have said. Yeah, people have been living and working with living things like, since the get-go. I mean, if you want to go all the way back, there's of course a number of theories about the nature of human civilization and democracy that rest on domestication as a core event. So I mean, if you take that broad view, you could definitely say that these types of relationships, the types of knowledge that people have to make them, has played a continually present role in the development of human society.
And this is the last session.
Yeah.
What is maybe a big lesson that we learned from all this beautiful history that might best inform us?
I think it's that you need to be very aware of the uses and the ends to which particular technology is going to be used. Like there's a number of things, for example, in biomanufacturing I might talk about in terms of making new therapeutics. But there's a very fierce debate that's happening right now. Is that actually something that's more useful than the alternative? Or is it something that's simply being done to satisfy, say, a patent cliff or some other reason that an individual company might have? And I think that's exactly the big, like there will be many more innovations going forward, and I'm in the wrong department to speak about those with any expertise. But I can say that there are often and always going to be alternatives. And those alternatives are going to be the thing to weigh as we sort of decide which of those innovations are worth investing heavily in, at least from the perspective of advancing social well-being. So that would be my slightly crotchety, but hopefully forward-looking answer.
Wonderful way to end. Thanks so much again for a great seminar.
Yeah, thank you.
Thank you.